Hi, my name is Zach. I'm presenting a research that I've been working on for the past year and a half. It's called PerturbODE. It is an interpretable neural ordinary differential equations architecture for gene regulatory network discovery. And I've been working on this project under the supervision of David Knowles and Elham Azizi. So for us, gene regulatory networks describe the activation and repression relationships between genes through transcription factors. And as we know that these gene regulatory networks govern nonlinear dynamical systems. Our primary motivation is to develop a biologically realistic and large-scale method to discover gene regulatory networks from single-cell RNA-seq datasets with perturbations. Traditionally, people used to manually curate these GRNs using fluorescent or ChIP-seq experiments. These methods are causal or mechanistic. However, they're very labor-intensive. And the large-scale methods will directly compute the covariance between the genes. And these methods are computationally efficient, but they are not causal. And recent effort has been trying to get the best of the two worlds. Most of the recent methods consists of causal differential graphical models with acyclic constraints, meaning the network that they discover is not cyclic. The linear models include node tiers and a low-rank version of node tiers. And there are also neural network-based models such as DCDI, which is currently, in my opinion, the state of the art. Nevertheless, it's not very scalable, and it can only handle up to 400 genes in the improved version. And therefore, there's a low-rank version of DCDI, which is the DCDFG. And it's much scalable, but its performance is a lot worse than DCDI. Therefore, and our second motivation is to model cellular trajectories in response to genetic perturbations. And most of the current methods involve optimal transport and flow matching. And as we know, that optimal transport and flow matching learn vector fields that are gradient or straight lines. And this is not very realistic because gradient systems or straight lines will prohibit cell cycles. And as we know that cell cycles are a very important phenomenon in biology. Further, these methods can only model one perturbation at a time, meaning you will learn one specific model for one perturbation only. Therefore, we're presenting our method, PerturbODE. It uses dynamical systems to model velocity in the gene expression space. As you can see here, the right-hand side of this equation parameterizes a vector field according to which cells will flow in the gene expression space. This vector field consists of three major components. First, a two-layer MLP, a linear decay term, and a perturbation term. So the two-layer MLP will encode the gene regulatory network, and it would also capture the regulatory relationship between the genes. And the linear decay term will model the degradation of mRNA molecules. And the perturbation term is basically a scaled standard basis vector that we use to model the genetic overexpressions. So for each perturbation, we will push forward the initial distribution or the control group in this vector field before comparing it to our target distribution and backpropagate using the Wasserstein 2 distance. It is important to point out that the two-layer MLP and the linear decay are shared among all the perturbations, and we will manually set the standard basis vector to specify which genes we are intervening on. Our gene regulatory network is obtained by essentially just multiplying the two-way matrices of the MLP. And the hidden neurons are analogous to gene modules that could potentially capture biological pathways. The real data set that we look at is the TF Atlas. It is a single-cell RNA-seq data set with 1,800 transcription factor overexpressions that are observed 7 days after perturbations. And these are human embryonic stem cells. And we also have a control group, which is the M-cherry. We imposed a L1 regularization only on the first weight matrix of the two-layer MLP. This is in accordance with the biological prior of network patterns in yeast and E. coli that the multiple output feed-forward loops are a lot more common than the multiple input feed-forward loops. We also have an additional diffusion-based contraction mapping we also have an additional training objective of diffusion-based contraction mapping. So we would diffuse our target distributions before shrinking it back. This would create a contraction map that leads to stable fixed points. It is biologically realistic to expect cells to stay at the basin of attractions over extended periods of time. So basically, by better modeling the biological nature of this process, we hope to better capture the causal mechanism of the gene regulatory network. We first experimented our method on simulated data, where we simulated some data using by sampling from stochastic differential equations with a known yeast GRN with 400 genes and 10 randomly generated acyclic GRNs with 100 genes. And as you can see here, that we outperform every other method except for DCDI, which is currently the state of the art. But the problem with DCDI is that it's not very scalable. And on the real data set, we outperform all the other methods, and DCDI doesn't scale to the real TF Atlas dataset with almost 1,000 genes. And we obtained the recall score by benchmarking against the known GRN edges. And we also obtain a p-value by comparing to random matrices. In fact, our method obtains better recall score at all sparsity levels comparing to other methods. And the different sparsity levels are obtained by applying different thresholds in classifying the edges. PerturbODE is also capable of accurately predicting response to unseen interventions. So we held out 10 TF overexpressions for testing, and we compare ourselves against 2 linear SCMs, no tiers and the low rank version of no tiers. Clearly, we outperformed the 2 other methods, both in terms of Wasserstein-2 distance and Pearson correlation. So for each one of the held out TFs, we can see the Wasserstein-2 distance over here, and 7 out of 10 times we outperform the other 2 methods. But as you can see here, when we do actually mispredict, we tend to miss by a lot due to the dynamic nature of our method. And if we visualize our prediction using UMAP, we can see that so the purple ones are the initial distribution, and the red one is our prediction, and the blue points are the target. As you can see here, that we clearly outperform the other 2 methods in terms of UMAP. To assess the stability of our model, we bootstrap the training data set and perform a stability analysis. And as you can see here, the known GRN edges are consistently selected by our model, and they are selected around 50 to 70% of the time. We also conducted a power analysis. And as you can see here, as we increase the number of perturbations included for training, our model performance steadily increases. And this is in accordance with our expectation. We also conducted a gene enrichment analysis that shows clustering of modules for different pathways. So we can, in fact, extract the gene modules for the ground truth GRNs that specify various different cell types by computing a score of the correct downstream and upstream genes in the modules. And if we specifically look at the gene enrichment analysis on these selected modules, we can see that these modules are indeed enriched in pathways related to their respective cell types. So in conclusion, PerturbODE is a highly scalable and biologically realistic approach to discover gene regulatory networks with thousands of genes from single-cell RNA-seq datasets with perturbations. In the future, we hope to extend our method to train on datasets with multiple time points, as well as incorporating ChIP-seq and ATAC-seq data as additional constraints and regularizations to narrow the scope of edge candidates. And this can especially reduce the false discovery rate by having more constraints. And thank you so much for listening. Please send me an email if you have any questions or suggestions. And I'm very happy to hear your feedback on this project.